Good morning, everyone. Welcome to Get Ahead Institute, where we transform potential into success. In this lecture, we will look at renal physiology. Specifically, we'll talk about renal function and micturition. So let's get into this. So to just introduce what renal physiology is all about. When we talk about renal physiology, or when we say the urinary system, this system or renal physiology is made up of the kidneys, ureters, urinary bladder, and the urethra. Okay, so now when we talk about the functions, um, when we talk about the functions of this renal physiology, or when we talk about the renal system, it has to do with excretion of waste products. Okay, so removal of wastes, for example, drugs and urea. And then also the other function is that it has to do with homeostasis. Okay, so under homeostasis, that is where we have um, electrolyte balance. Okay, it has to do with electrolyte balance. It has to do with water balance. It has to do with the control or the regulation of blood volume. Okay, and then another function is that it is involved in regulation of number one, blood pressure. Okay, though we've not just included it here on the slides, blood pressure, um, acid base balance. Okay, it has to do with bone health. That is where we link it to calcium levels. Okay, it has to do with hemopoietic balance. Um, Okay, so those are just the functions of uh, the urinary system or the renal system. Okay, so we've already said that this urinary system is made up of the kidneys, ureters, urinary bladder, and the urethra. Okay, so you need to understand that when we are talking about the renal system, blood basically enters the renal system via the renal artery, and then it leaves through the renal vein. And then you find that blood is filtered by nephrons. We'll talk about the nephrons in the next few slides as we are proceeding. Okay, and then you will find that urine is basically transported from the kidney by the ureter to the bladder, and then it is stored in the bladder before it is excreted. Okay, so one of the things that you need to take note of is that the composition of blood is different. That is the blood that is entering and that which is leaving the kidneys. For example, you'll find that the renal vein basically has number one, it has reduced or it has a decrease in water and solutes. Number two, it has a decrease in urea. It has a decrease in glucose. It has a decrease in oxygen, but it has some abundance of carbon dioxide. Okay, that is with regards to the renal system as far as the composition of blood is concerned when we compare it with that which is coming in and that which is leaving. All right. So this is the nephron. Okay. So this is the nephron. This is basically the nephron. So when we talk about the nephron, by definition, the nephron is just the structural and functional unit of the kidney, and we have approximately um, 2.4. Okay, we have approximately 2.4 million. Okay, we have approximately 2.4, or we can say 1.3 million to 2.4 million um, of nephrons collectively in both kidneys. Okay, collectively in both kidneys. When we talk about this nephron, Okay, when we talk about this nephron, the nephron basically is composed of the Bowman's capsule. Okay, we have the Bowman's capsule, we have the glomerulus. Okay, so when we combine the glomerulus and the Bowman's capsule, we make what is known as the renal capsule. Okay, we make what we call the renal capsule, which is this one. So, in other words, the renal capsule is just a combination of the Bowman's capsule and the glomerulus. Okay, and then we have the renal tubule. Okay, we have the renal tubule. So this renal tubule basically we'll look at at it in detail later. Okay, so the renal tubule basically, when you look at the renal tubule, um, this functionally it is divided into segments. We have the proximal convoluted tubule, we have the loop of Henle, and we have the distal convoluted tubule. Okay, so basically, the nephron is composed of renal corpuscle, and we have a long tubule, okay, which is divided into three proper segments. And then when we talk about this um, nephron, we have types of nephrons. We have what is known as the cortical or superficial nephrons. Okay, we have the cortical or the superficial nephrons. So these ones, these are nephrons having the corpuscles in the outer cortex, just near the periphery. And then we have the juxtamedullary nephrons. So these nephrons basically have um, uh, they have in they have or we can say that when we compare them to the cortical nephrons, basically these ones they are closest to the medulla. Okay, they are closest to the medulla, and they have long loops of Henle. Okay, which basically just extend down into the medullary pyramids. So um, those and by the way, when we talk about regional differences in the kidney, when we talk about regional differences in the kidney, we have the renal cortex, which is just the outer portion and it contains the renal corpuscles. And then we also have the renal medulla, which is just the inner portion. Okay, this is just the inner portion. All right. So apart from these cortical and juxtamedullary, we have this one which is rarely talked about. We have the mid cortical nephrons. Okay, we have the mid cortical nephrons, or we can say intermediate nephrons. Okay, so now you need to understand one thing that in humans, okay, about in humans about 7 out of 8, okay, about 7 out of 8 in humans, all nephrons are cortical. Okay, all nephrons are cortical nephrons, and then 1 out of 8, okay, 1 out of 8, they are basically juxtamedullary nephrons. So we have more of these nephrons. Okay, we have more of the cortical nephrons compared to the juxtamedullary nephrons. Okay, so now when we talk about the structure of the nephron, you need to understand that the nephron basically is comprises of the Bowman's capsule. Just like we mentioned earlier, we have the Bowman's capsule. Then we have let me just take you to the slide. Let me just take you to this slide. Okay. So this is the structure of a nephron. Okay. So you'll find that you have the Bowman's capsule. So when it comes to the Bowman's capsule, remember earlier we said that when you combine the Bowman's capsule and the glomerulus, okay, so the glomerulus, it is this which is inside. So this glomerulus is basically just inside the Bowman's capsule, okay, inside the Bowman's capsule. And then we have the proximal covulated tubule, okay, we have the proximal covulated tubule right here, okay. So when you are coming like that, the proximal convoluted tubule, then you start, you find we have the loop of Henle. Okay, so this is the loop of Henle right there. Specifically, this is the descending limb of the loop of Henle, and then you have the ascending limb of the loop of Henle. Okay, then from there we have the distal convoluted tubule. Okay, we have the distal convoluted tubule, which is right here, and then we have other literature will also mention before we talk about the collecting duct. It other literature will say, or other books say, that we have the collecting tubule. Okay, so the collecting tubule basically just connects the distal tubule to the collecting duct. Okay, so from the collecting tubule we have the collecting duct right there. Okay, so this is the structure of a nephron. So when we talk about collecting ducts, okay, when we talk about collecting ducts, the epithelium of the collecting ducts basically comprises of the P cells. Okay, it has P cells and the I cells. So when we talk about the P cells, which are the principal cells, these principal cells are basically involved in sodium reabsorption and vasopressin stimulated water reabsorption. Okay, and then when it comes to the eye cells are just concerned with acid secretion and um, this other component is transport. Okay, when it comes to collecting ducts. So from there we move on to renal circulation. So under renal circulation, you need to understand one thing, that when we're talking about this renal circulation, then we're talking about, we've now delved into what we can say that we're talking about the nephron and its blood vessels. Okay, the nephron and its blood vessels. So you'll find that we've already said you need to understand that blood basically enters the glomerulus via the afferent arteriole. Okay, blood enters the glomerulus via the afferent arteriole. Okay, so blood will enter the glomerulus via the afferent arteriole, and then it will leave via the efferent arteriole. Okay, and then those efferent arterioles will then drain into number one, the peritubular capillaries. Okay, peritubular capillaries. So these capillaries basically just surround the tubules of the nephrons, and then they'll also drain into number two, vasa recta. Okay, will also drain into vasa recta. So these vasa recta, they basically these are the ones which surround the loop of Henle, specifically of the juxtaglomerular nephrons. Okay, so when it comes to renal circulation, you need to understand that blood will basically move from, we have the abdominal aorta, we have the renal artery basically in the renal pelvis, and it will move to interlobar arteries, and then it will move to corticomedullary junction, which are just specifically the arcuate arteries. Then from there, we'll have interlobular arteries, which will proceed to afferent arteries. Then from there, would have reached now the glomerulus. Then from the glomerulus, we are going to have the efferent artery. Okay, and then from the efferent artery, that is when now we are going to have the peritubular capillaries. Then from the peritubular capillaries, we are going to have the intertubular veins. From there, we'll have arcuate veins, which will lead us into the interlobar veins, and then finally we'll end with the renal vein. Okay, so we are saying all this before we talk about the blood flow, the renal blood flow. Okay, so you'll find that when we talk about vasa recta, these vasa recta basically have different structures along their tracts. Okay, so you'll find that along the descending, you'll find that they are like on the descending loop, you'll find that they are more like arterial, like non-fenestrated endothelium, which basically contains a facilitated transporter of urea. Then around the loop, they are more like capillary like. Okay, then on the ascending aspect, they have fenestrated endothelium in order to allow solute exchange and conservation. Okay, then now from there we can talk about the renal blood flow. Now we talk about renal blood flow. Okay, all right, so now you need to understand one thing, that as far as renal blood flow is concerned, in a resting adult, usually the kidneys only, okay, the kidneys only receive 1.2 to 1.3 liters of blood per minute. Okay, or we can just say below or under 25% of the cardiac output. And then you need to understand that usually the kidney filters the plasma. Okay, so you'll find that the renal plasma flow is normally equal to the amount of a substance which is excreted per unit time, then thereby divided by the renal arteriovenous difference, so long as the amount in the red cells is unaltered during the passage through the kidney. Okay, so since we're talking about renal, we're still talking about we're talking about blood flow. Okay, we're talking about blood flow. You need to understand that I have put these important concepts here deliberately, very very important, but I'll try to explain them. So we have the P amino hippuric acid, PAH. Okay, so what is this PAH? Okay, so you need to understand that now I want us to talk about renal plasma flow. Okay, so when we're talking about renal plasma flow, this renal plasma flow basically can be determined, or it can be measured by infusing para aminohippuric acid and later determining its urine and plasma concentrations. Okay, so you'll discover that this PAH is usually filtered by the glomeruli and then secreted by the tubular cells, so that its extraction ratio. So when we're talking about extraction ratio, which is right here, when we're talking about this extraction ratio, this is just arterial concentration minus renal venous concentration divided by arterial concentration. Okay, very very important to take note of that. Okay, so now um you need to understand that it has now therefore become a very commonplace to calculate renal plasma flow just by dividing the amount of the PAH in the urine by the plasma, okay, by the plasma PAH, whereby we now ignore the level of renal venous blood, okay. But you need to understand that when it comes to the peripheral venous plasma, so the peripheral venous plasma can be used because its PAH concentration is essentially identical to that in the arterial plasma that is reaching the kidney. So now, after you've done those calculations with what we have what I've said so far, the value that the value that has been obtained is what is known as the effective renal plasma flow. Okay, effective renal plasma flow. Okay, so this effective renal plasma flow basically just indicates that um, the level that the this one when we measure the effective renal plasma flow, it is just there to indicate that the level in the renal venous plasma was not measured. Okay, so it's very very important also to understand and to take note that when it comes to this effective renal plasma flow in humans, it is about 625 milliliter per minute. Okay, so this is the value. So if you want to calculate effective renal plasma flow, this is the formula that you use. Okay, this is the formula that you use. So this U stands for urine. Okay, this U stands for urine, PAH. Then this is the rate. Okay, the flow rate. Okay, this is for the flow rate. Okay, so this is for the urine flow rate, and then you have this is the plasma. This is PAH, just like this one. So what you get, you can term it as the clearance of PAH. Okay, the clearance of PAH. Let's look at this example. So in this example, we are being told that given the concentration, okay, given the concentration of PAH in urine, okay, PAH in urine to be 4 milligram per milliliter, then the urine flow rate, okay, urine flow rate is just 9 milliliter per minute, and also been given the concentration of PAH in plasma, which is just 0.02 milligram per milliliter. They want us to calculate this. They want us to find renal plasma flow. Okay. So in order to calculate renal plasma flow, what will happen is we're going to use the very formula. Okay. So we're going to say because we want us to find renal plasma flow, and you can't just jump just like that. And yeah, so what will happen is we have effective renal plasma flow. Okay, we have effective renal plasma flow, which will be equal to um, remember the formula which is on top right there that I showed you earlier. So I'll just replace the figures directly. So we're going to have the urine PAH, which is 14, okay, 14, and we are going to multiply it with the urine flow rate. So urine flow rate was just 0.9. Then we are going to divide with the pAh of plasma, which we are given to be 0.02, like that. Okay, so you'll find that when you get your calculator and punch, you definitely get 630. Okay, 630. The units for this are milliliter per minute. Okay, so this value that we've obtained is for effective renal plasma flow. Okay, the question is telling us to find the renal plasma flow. Okay, but now this one is effective, so you need to know how to solve this thing. Okay, so now it should be noted that the effective renal plasma flow, which has been determined this way, the way we've solved here, is the clearance. Okay, so this is just the clearance of PAH. Okay, this is just the clearance of PAH. Okay, so you will find that effective renal plasma flow can then be converted to actual renal plasma flow. Okay, actual renal plasma flow. Okay, so remember, always remember that the renal extraction ratio, okay, the renal extraction ratio of PAH in a normal individual is approximately 0.92, okay, but Ganon will put it as 0.9. Okay, that is just a figure, just need to know it. Okay, just need to know it. Okay, so now when we want to convert this effective to the actual renal plasma flow, okay, the actual renal plasma flow, what are we going to do? So what we are going to do is we are going to divide the effective, okay, we're going to divide the effective renal plasma flow, which is just 630 divided by the extraction ratio. Okay, so we're going to divide with the extraction ratio, which is just 0.9. Okay, like that. So you find that when you do the division, you basically get 700. Okay, you get 700, and the units for this are the milliliter per minute. Okay, so this is not our remember, we want to find the renal blood flow. Okay, this was just a typing error. We are supposed to find renal blood flow here. Yeah, like that. So let's find renal blood flow. So you'll find that after this value that we found, this is the renal plasma flow. So now this is the actual. Okay. Hope you followed the way we've moved. This is the actual. This was the effective, which is just the clearance of PAH, and then I wanted the actual one. So to get the actual, we divided the this value into the 630 that we have, and we got 700. Okay, so now for us to get from the renal plasma flow, the renal blood flow can be calculated by dividing by dividing by 1 minus the hematocrit. Okay, minus the what? The hematocrit. Okay, so remember, normal hematocrit basically for a normal human being in this case is 0.45. Okay, 0.45. So what will happen is our formula in order to find renal blood flow, okay, renal blood flow, it will be equals to um, renal plasma flow, the actual one, okay, divided by 1 minus hematocrit, okay, divided by 1 minus hematocrit, like that. So this is the formula for us to find renal blood flow. So when you just replace the figures, replace here you put 700, here you put 0.45, you get something like 1273. The units are milliliter per minute. Okay, so that is how we solve this. Okay, now when we talk about regulation, okay, when we talk about regulation of renal blood flow, we have things like norepinephrine. Okay, so you find that this norepinephrine basically it just constricts the renal vessels, with the greatest effect of the injected norepinephrine being exerted on the interlobular arteries and the efferent arterioles. Then we also have dopamine. Okay, then we also have dopamine. So this dopamine basically, what it does is that dopamine is made in the kidney and it causes renal vasodilation and natriuresis. Okay, causes vasodilation and natriuresis. Okay, and then we also have angiotensin 2. So angiotensin 2 just exerts a constrictor effect on both the afferent and the efferent arterioles. Then we also have prostaglandins. Okay, so these prostaglandins just increase blood flow in the renal cortex and decrease blood flow in the renal medulla. Then when we talk of acetylcholine, your favorite. So when we talk about acetylcholine, acetylcholine also produces renal vasodilation. Okay, and then when we have a high protein diet, so a high protein diet basically just raises the glomerular capillary pressure and increases renal blood flow. Okay, then when we talk about renal autoregulation, okay, when we talk about renal autoregulation, there this renal autoregulation is very important in the sense that it helps to maintain the glomerular filtration rate. Okay, it helps maintain the glomerular filtration rate. Okay, so let's start by talking about because there are two main mechanisms as far as renal autoregulation is concerned. There are two mechanisms. We have the myogenic response. So this myogenic response basically it has to do with this is an intrinsic mechanism. Okay, it is an intrinsic mechanism that regulates RBF, and it occurs in the afferent arterioles, which are just the small blood vessels which supply the glomeruli. So what happens is when blood pressure increases, the arterioles will constrict in order to reduce blood flow. And when the blood pressure decreases, the arterioles dilate. Okay, the arterioles will dilate, and when they dilate, they will increase blood flow. So you'll find that this response, which is known as myogenic response, is very, very important as it helps maintain a relatively constant renal blood flow despite the changes in systemic blood pressure. Then, secondly, we have tubular glomerular feedback. We have tubular glomerular feedback. So, as this one regulates based on the amount of sodium chloride in the tubular fluid. Okay, so this one is determined by the amount of sodium chloride in the tubular fluid. Okay, and it occurs in the juxtaglomerular apparatus where the distal convoluted tubule contacts the afferent arteriole. So what happens basically is that when sodium chloride levels in the tubular fluid increase, we have cells which are known as the macula densa. Okay, so the macula densa cells will detect and then send the signals to afferent arterioles to contract. And when the sodium chloride levels decrease, the macula densa brings about the dilation of the afferent arterioles. So basically, this feedback loop, which is the tubular glomerular feedback, it helps maintain a stable glomerular filtration rate, and it prevents um, excessive sodium loss in the urine. Okay, so we have adenosine. Okay, so adenosine basically just lowers glomerular filtration rate, okay, by constricting afferent arterioles, and on the other hand, renin increases glomerular filtration rate. And then I can also talk about though I didn't add it in my notes, but I can just talk about it. We have things to do with secretion of hormones. Okay, secretion of hormones. So you will find that the juxtaglomerular apparatus secretes two main major hormones. We have renin and prostaglandin. So what happens is renin, okay, what happens is renin basically along with angiotensin, renin forms the renin-angiotensin system, which helps maintain blood pressure. Okay, so the secretion of renin is basically stimulated by four factors. Number one, when there is a fall in arterial blood pressure, renin will be secreted. When we have a reduction in the ECF volume, renin will be secreted. When the sympathetic activity increases, okay, when sympathetic activity increases, renin will be secreted. And then when we have a decrease, okay, when we have a decrease in the load of sodium and chloride ions in the macula densa, okay, renin will be secreted. So now, what are the functions of angiotensin? So angiotensin 1, we know that it is physiologically inactive, okay, and it just serves as a precursor of angiotensin 2. And then it is now this angiotensin 2 which is the most active form, and its actions include: number one, this angiotensin 2 increases the arterial blood pressure by directly acting on the blood vessels and causing vasoconstriction. So angiotensin 2 is a very, very potent vasoconstrictor in arterioles. Number two, it increases blood pressure by indirectly increasing the release of noradrenaline from the postganglionic sympathetic fibers. Okay, so you need to take note that noradrenaline is a general vasoconstrictor, while angiotensin 2 is a potent vasoconstrictor of the arterioles. Okay, so we've talked about this. we've So now let's talk about the formation of urine. Okay, let's talk about the formation of urine. So urine, normal urine output is supposed to be 1 to 1.5 liters per day. That is the normal urine output, 1 to 1.5 liter per day. And this process of urine formation involves three main processes. Okay, we have number one, it involves glomerular filtration. Then it also involves tubular reabsorption and tubular secretion. Okay, so let's begin by talking about glomerular filtration. So when we talk about glomerular filtration, this is just the amount of plasma ultrafiltrate which is formed each minute. Okay, so what happens is that blood is normally filtered while passing through the glomerular capillaries by filtration, by the filtration membrane. Okay, and then you need to understand that the structure of the filtration membrane is well suited for filtration as it is formed of three main layers. Number one, it comprises of the glomerular capillary membrane. Number two, it has a basement membrane. Number three, it has the visceral layer of Bowman capsule. So you also need to take note that glomerular filtration is also known as ultrafiltration simply because even the minute particles are basically filtered, okay? But then the plasma proteins are not allowed because they have very large molecular size, okay? So is therefore you need to understand that the filtrate, okay, the ultrafiltrate which is formed contains all the substances in plasma except the plasma proteins. Okay, so now back to glomerular filtration rate, of which we've just said that this is just the total quantity of the filtrate which is formed in all the nephrons of both the kidneys in the given limb, I mean the given unit of time. Okay, so ideal glomerular marker, the ideal glomerular marker is inulin, okay? Is inulin, okay? This is the ideal glomerular marker, inulin, okay? So the normal glomerular filtration rate of a normal person, of a normal adult is 125 milliliter per minute, or we can say about 180 liter per day. And then we have a formula that you can use in order to calculate glomerular filtration rate. That is just glomerular filtration rate is just equal to the concentration of urine. Okay, so the concentration of inulin in urine. Okay, the concentration of inulin in urine multiplied by flow rate divided by the concentration of inulin in plasma. Okay, this is the formula if you want to calculate glomerular filtration rate. Okay, so this is known, what you find here is what is known as the clearance of inulin. Okay, clearance of inulin, like that. Okay, and then now kidneys. Okay, kidneys Under normal circumstances, under normal cardiac cycle, they just receive under 25% of the normal cardiac cycle. I think I already mentioned that. Okay, so we have filtration fraction. Okay, so a filtration fraction, when you talk about the filtration fraction, let me just paraphrase what is on this very slide. This just has to do with the portion of renal plasma which becomes the filtrate. Okay, so you will discover that the ratio between renal plasma flow and glomerular filtration rate is expressed in a percentage. Okay, and it is normally between 0.16 to 0.20, which is just 16% to 20%. That is the filtration fraction. Okay, then if you want to calculate filtration fraction, is just dividing glomerular filtration rate divided by renal plasma flow multiplied by 100. Okay, then another way you can calculate renal plasma flow. Another way you can calculate renal plasma flow, let me just find where I can write from. Let me just use this very space. So another way you can calculate renal plasma flow, renal plasma flow will be equal to um, the concentration, like that concentration of PAH. Okay, so this will be concentration of PAH basically um, in urine multiplied by the flow rate, and then you divide by concentration of this is renal artery. Okay, renal artery PAH. So here we'll put PAH and then minus concentration of renal vein. Okay, so we are talking about, for you to find renal blood flow, it can be concentration of PAH in urine multiplied by flow rate divided by concentration of PAH in renal artery minus concentration of PAH in renal vein. Then you get your renal plasma flow. So now there are pressures. Okay, we have certain pressures. We have certain pressure which basically we have certain pressure which we have certain pressure which normally determine filtration. Okay, by the way, normal renal plasma flow is 600 milliliter per minute. Now we have pressures which determine um, glomerular filtration rate. Number one, we have the glomerular capillary pressure. Then we have the colloidal osmotic pressure in the glomeruli. Then we also have hydrostatic pressure in the Bowman capsule. When we talk about net filtration pressure, this is just the balance between the pressure favoring filtration and the pressures opposing filtration. Okay, so for you to find the net filtration pressure, it is just glomerular capillary filtration minus colloidal osmotic pressure plus hydrostatic pressure. So usually the net filtration pressure is somewhere approximately 20 millimeter of mercury. Okay, so we have some factors here. um, so these are factors which affect or which regulate glomerular filtration rate. Okay, for example, renal blood flow increases glomerular filtration rate. Tubulo-glomerular feedback decreases glomerular filtration rate. Then glomerular capillary um, pressure, it it increases glomerular filtration rate. And then colloidal osmotic pressure decreases glomerular filtration rate. And then hydrostatic pressure decreases glomerular filtration rate. Even hormones, they also have effect on the glomerular filtration rate. Remember, we are talking about urine formation. We've talked about that is about um, filtration. Okay, that is about filtration. Now let's talk about tubular reabsorption. That was about glomerular filtration. Now let's talk about tubular reabsorption. So when we talk about tubular reabsorption, you need to understand that water and other substances are usually transported back into the blood from the renal tubules. And the routes of reabsorption include transcellular and paracellular. Okay, so we have sites, we have specific sites for reabsorption. Okay, we have specific sites for reabsorption. We have the proximal convoluted tubule. So on the proximal convoluted tubule, things which are reabsorbed include glucose, amino acids, sodium, potassium, calcium, bicarbonate, chloride, phosphate, urea, uric acid, and water. Then we have the loop of Henle. Okay, so the loop of Henle, things which are reabsorbed there, we have sodium and chloride. Then we have the distal convoluted tubule. Here, things which are reabsorbed include sodium, calcium, bicarbonate, and water. Okay. Then we proceed. So we have already talked about all these. So these are factors which regulate tubular reabsorption. We have the glomerulo-tubular balance. Glomerulo-tubular balance. Okay, glomerulotubular balance. And we have the hormonal factors. We also have the nervous factors. Okay. From there, we can talk about tubular secretion. So these are the processes. Okay. We can talk about tubular secretion. So this, when we talk about tubular secretion, has to do with substances which are secreted from the blood to the renal tubules. Okay. So the secreted substances include things like the para-aminohippuric acid, you have the amino derivatives, you have some drugs which include penicillin. Okay. Then we come to concentration of urine. Okay. Remember, I'm talking about renal function and micturition. Okay. So now, before urine can be excreted, it needs to undergo some process of concentration. Okay. So now, when we talk about concentration of urine, normally urine is concentrated and its osmolarity is 4 times than the normal osmolarity. That is 1,200 milliosmoles per liter. Okay, and you need to understand that osmolarity of urine basically depends on two factors: the water content in the body and antidiuretic hormone. Now, we will talk about dilute urine formation, how we come up with dilute urine, the formation of dilute urine, and also concentrated urine formation. So let me start by talking about this aspect, which is dilute urine formation. So when water content in the body increases, the kidney excretes dilute urine. And this is achieved by the inhibition of antidiuretic hormone secretion from the posterior pituitary gland. Okay, so when water is a lot in the body, the kidney will excrete more, uh, a high volume of urine with less concentration. Okay, because ADH has been inhibited and it won't be able to bring about the absorption of water. Okay, then now, how do we form concentrated urine? How do we form concentrated urine? So you need to understand that forming concentrated urine is not, um, when we are talking about concentration of urine, usually it occurs when water content in the body decreases. Okay, so when water content in the body decreases, the kidney excretes concentrated urine. But it is important to take note that the formation of concentrated urine is not a simple process. It involves two major processes. It involves two major processes. We have number one, um, the process which has to do with the medullary gradient. Okay, medullary gradient. So the medullary gradient includes two aspects. You have the countercurrent multiplier. Okay, you have the countercurrent multiplier, which is by the loop of Henle. Okay, so now how does this countercurrent multiplier work? Okay, so by the way, when we're talking about the medullary gradient, this is also known as the osmotic gradient. Okay, so now I want us to look at how the countercurrent multiplier by the loop of Henle works. Okay, so this mechanism basically just works through the counter. When we're talking about the loop of Henle, how it creates a concentration gradient in the medulla through the countercurrent multiplier mechanism, we have the descending limb. Okay, so the descending limb of the loop of Henle, there you find that water and ions, that is sodium, potassium, and chloride, freely diffuse out. And when they are diffusing, they are increasing the interstitial osmolarity. Okay, and then the thick ascending limb, which is this one has to do with active reabsorption of sodium, potassium, and chloride ions without water, which further increases interstitial osmolarity. Then the thin ascending limb, okay, which is a thin ascending limb, has to do with passive reabsorption of sodium and chloride ions, maintaining the gradient. Okay, so you'll find that this process, which I've said, which involves the descending limb, the thick ascending limb, and thin ascending limb, basically creates a hyperosmotic environment in the medulla, which is essential for water reabsorption and concentrated urine formation. Then we have the counter current exchange by the vasa recta. Okay, remember the vasa recta, these are just capillaries which are surrounding the loop of Henle, which assist in maintaining the medullary gradient through the countercurrent exchange. Okay, so under the countercurrent exchanger, we have things to do with descending vasa recta. So under the descending vasa recta, we have things to do with oxygen and nutrients where they diffuse out while waste products diffuse in. And then the ascending vasa recta, we have oxygen and nutrients diffusing in while waste products diffuse out. So you discover that it is this, during this exchange, we have the preservation of the osmotic gradient, which minimizes washout of ions and solutes. Okay, and then it helps maintain the concentration of urea and other solutes in the medulla. So when we look at the combined effects of the countercurrent multiplier and the countercurrent exchanger, you discover that it there is an establishment and maintenance in the medullary gradient, okay? And this combined effect enables water reabsorption in the collecting duct and concentrating urine, okay? It also assists in, it comes in as it helps regulate electrolyte balance and acid-base homeostasis, okay? So when we are forming concentrated urine, you'll find that when antidiuretic hormone stimulates water reabsorption in the collecting duct, number one, water moves from the tubular lumen to the hyperosmotic interstitium, then urine concentration increases. Okay. So in summary, we can say that the countercurrent multiplier and the countercurrent exchange, they work in synchronicity or together in order to establish and maintain the medullary gradient, which enables the formation of concentrated urine. Then we in though I have talked about antidiuretic hormone, but let me talk about it separately as it also contributes to formation of concentrated urine. Okay, so secretion of antidiuretic hormone. You need to understand that the final concentration of urine is done by ADH. Okay, so normally the distal convoluted tubule and the collecting duct are not permeable to water. But now the good thing is that ADH makes them permeable, which results in water reabsorption. Okay, so the water reabsorption which is induced by ADH is known as facultative reabsorption. Okay, so one of the things, let me just take you back to the countercurrent. One of the things that we need to take note is that the countercurrent mechanism depends on increased osmolarity along the medullary pyramids and are produced by the countercurrent multipliers and then are maintained by the countercurrent exchangers. Okay, so these are just some things to help you just master these things. That when we talk about vasoconstrictors, these are vasoconstrictors that we have. Then dilators, these are dilators. Then we have what we call osmotic diuresis. Okay, we have what we call osmotic diuresis. Okay, so this osmotic diuresis, when we have the presence of large quantities of unreabsorbed solutes in the renal tubules, it brings about an increase in urine volume. Okay, it brings about an increase in urine volume, and we call that osmotic diuresis. Okay, so in other words, this osmotic diuresis can be described or defined as a condition or a situation where we have excessive amounts of osmotically active substances, for example, glucose, urea, in the renal tubules, begin to inhibit water reabsorption, leading to increased urine production. Okay, so now what is the mechanism of this osmotic diuresis? Okay, number one, we have increased osmotic pressure in the tubular lumen. Number two, inhibition of water reabsorption in the proximal convoluted tubule and collecting duct. Number three, increased water delivery to the distal nephron. Number four, impaired concentration ability of the kidneys, and what could be the causes of this osmotic diuresis. Number one, it can have to do with hyperglycemia, that is diabetes mellitus. We can have uremia. Okay, so uremia has to do with renal failure. Um, we can have mannitol administration, which are these are just diuretics. We can have glucose intolerance. Okay, as well as inherited disorders, for example, diabetes insipidus. Okay, so when it comes to osmotic diuresis, there are some characteristics which include polyuria. Okay, polyuria, which is just excessive urine production. Then we can also have it is also characterized by polydipsia, which is just excessive thirst, dilute urine. Okay, electrolyte imbalances, e.g., hypokalemia, as well as dehydration. Okay, because water is not reabsorbed. Thank you so very much. This is the end of this lecture.